Cnidaria is one of those scientific names that sounds narrow until you realize what it contains. Jellyfish blooms, reef-building corals, sea anemones in tide pools, freshwater hydra, drifting hydrozoan colonies, and even tiny parasitic forms all belong to this single phylum. That range matters because it tells us we are not looking at an obscure side branch of life. We are looking at a deeply successful design that has been reused in very different habitats. The core cnidarian plan is compact, 2 tissue layers, a jelly-like matrix called mesoglea between them, a central cavity with 1 opening, and tentacles loaded with stinging cells. At first glance this seems almost too simple compared with animals that have brains, hearts, and complex organs. But in evolution, simplicity is not failure. If a design keeps working while environments change for hundreds of millions of years, that design deserves attention. Cnidarians do not dominate through speed or intelligence in the human sense. They dominate by turning water flow, contact, and chemistry into reliable feeding and survival strategies with low biological overhead. The signature innovation of cnidarians is the cnidocyte, a specialized cell that acts like a microscopic launch system. Inside is a tightly packed organelle that can fire almost instantly when triggered, often by touch and chemical cues. Depending on the type, that discharge can inject venom, latch onto prey, or entangle it. This is central to cnidarian ecology because many species are not active chasers. They do not need to pursue prey over long distances when they can create a lethal interception zone around their body. Tentacles become functional nets, and cnidocytes provide the stopping power. For tiny prey like plankton, this can mean immediate immobilization. For larger targets, repeated contacts can overwhelm escape responses. The same system also discourages predators, so it doubles as defense. What makes this elegant is that predation, defense, and feeding all emerge from distributed cellular tools rather than expensive centralized anatomy. Cnidocytes are not just stingers. They are a modular technology that lets cnidarians convert passing opportunity into energy with remarkable efficiency. Cnidarian bodies are usually radially symmetrical, and that geometry fits aquatic life where signals and prey can approach from any direction. Instead of a clear head and tail, they organize around a central mouth opening ringed by tentacles. This architecture supports 360-degree interaction with the environment, which is ideal for drifting or anchored animals in moving water. Internally, many cnidarians rely on a single cavity for digestion, with 1 opening used for both intake and waste release. By vertebrate standards that sounds crude, but with thin tissue layers and short diffusion distances, it can work effectively. The mesoglea between layers contributes structural support and, in medusae, helps with buoyancy and elastic movement. The overall design reduces maintenance costs because fewer specialized organs need constant energy input. That low-cost baseline can be a major advantage in nutrient-variable environments. So radial symmetry and a single opening gut are not random leftovers from early evolution. They are part of a coherent strategy, maintain broad environmental responsiveness while keeping metabolic demands manageable. Most introductions to cnidarians emphasize 2 body forms, the polyp and the medusa. The polyp is usually attached to a surface, with tentacles positioned to capture food from passing currents. The medusa is free-swimming, often bell-shaped, using rhythmic contractions to move through the water column. These are not minor variations. They represent different ecological modes. Polyps are excellent at holding territory, cloning, and building local persistence. Medusae are excellent at dispersal, gene flow, and reaching new habitats. Many cnidarians alternate between these forms during their life cycle, effectively switching survival tactics across generations. Some lineages reduce 1 stage, and a few abandon the classic pattern altogether, but the broader point remains. Cnidarians can combine station keeping and mobility within one evolutionary framework. That flexibility helps explain their wide distribution from shallow coasts to open ocean systems. A lineage that can anchor when conditions are good and disperse when conditions shift has a built-in hedge against environmental instability. Their reproductive strategy is similarly flexible. Asexual reproduction, often through budding and polyp stages, allows rapid local expansion without waiting for mates. If a genotype is well suited to current conditions, cloning can quickly build population density and, in some groups, create integrated colonies. Sexual reproduction introduces recombination, generating variation that can improve resilience when conditions change or new pressures emerge. Cnidarians often combine both approaches in one life cycle, using each mode where it performs best. This dual system matters over long timescales. Cloning provides short-term efficiency and ecological occupation. Sexual cycles provide long-term adaptive potential. Some taxa streamline this pattern by minimizing one phase, while highly derived parasitic cnidarians diverge so far that they no longer resemble familiar jellyfish or coral forms. Even then, they remain part of the same evolutionary story. The key insight is that cnidarians are not locked into one demographic rhythm. They can shift between replication strategies, balancing speed, stability, and adaptability across changing environments. A frequent misconception is that cnidarians are behaviorally primitive because they lack centralized brains. In reality, they use decentralized nerve nets that coordinate contractions, feeding responses, and orientation across the body. Signals can propagate through distributed neural pathways, allowing local information to produce whole-body responses without a single command center. This fits their radial organization, where no permanent forward direction dominates. Some groups add specialized sensory structures. Rhopalia can support gravity sensing and chemical detection, statocysts assist with balance, and certain box jellies and true jellyfish possess simple eyes that provide useful visual cues. These systems do not create mammal-style cognition, but they do provide enough environmental awareness for effective navigation, prey capture, and threat response. The lesson here is important. Centralized intelligence is only one route to functional behavior. Cnidarians show that distributed control, when matched to body design and ecological niche, can be highly effective. They survive not by thinking like vertebrates, but by integrating sensation and action in a way that is energetically lean and ecologically sufficient. The phylum's major classes illustrate how far one blueprint can diversify. Anthozoa includes corals, sea anemones, and sea pens, mostly emphasizing polyp forms and often sessile lifestyles. Scyphozoa contains many familiar jellyfish that dominate pelagic visuals in public imagination. Cubozoa, the box jellies, are especially notable for potent venom systems and relatively advanced sensory organization. Hydrozoa is broad and flexible, including freshwater hydra, marine-attached forms, and colonial drifters such as Portuguese man-o'-war. Storozoa has been elevated as a distinct class rather than treated as a minor subgroup, reflecting improved phylogenetic resolution. Then there are extreme cases like Myxozoa and Polypodiazoa, parasitic lineages so reduced that their cnidarian identity was confirmed only with modern analysis. Together, these groups show that Cnidaria is not a single lifestyle category. It is an evolutionary platform that supports attachment, drifting, active pulsing locomotion, colony integration, and parasitism. Diversity here is not despite the simple core plan. It is enabled by it. Colonial cnidarians are especially useful for challenging our intuition about what counts as an individual organism. In many colonies, repeated units called zooids specialize in different functions such as feeding, reproduction, or defense, yet remain physiologically integrated as one organismal system. To an observer, this can look like a community, but biologically it behaves more like a distributed body made of repeated modules. This arrangement allows division of labor without requiring each unit to perform every task. Functional specialization can improve efficiency, especially in variable flow environments where feeding opportunity, predation risk, and reproductive timing shift over time. Coloniality also enables size and complexity gains while retaining clonal genetic continuity. In practical ecology, colonial forms can occupy space effectively, intercept more prey, and withstand partial damage because functions are distributed. The broader biological message is that cnidarians blur boundaries between singular and collective life. They reveal that evolution can build integrated systems from repeated simpler units, creating emergent performance without conventional organ-level complexity. Coral-associated cnidarians demonstrate another level of integration through symbiosis with photosynthetic dinoflagellates, commonly called Zooxanthellae. In reef-building corals, these symbionts live within host tissues and provide energy-rich compounds generated from sunlight. The coral, in turn, provides habitat and access to light and dissolved nutrients. This partnership transforms ecological potential. It helps corals thrive in tropical waters that can be relatively low in free nutrients, and it underpins reef growth that supports extraordinary biodiversity. But symbiosis is a trade-off as well as a strength. When environmental conditions strain the partnership, corals can lose symbionts, reducing energy input and compromising growth or survival. Even without diving into every climate variable, the biological pattern is clear. Collaboration across species can multiply productivity while increasing dependence. For cnidarians, this means some of their greatest ecosystem achievements are inseparable from microbial partnerships. Reef systems are therefore not just coral structures. They are metabolic alliances operating at massive spatial scales. Cnidarians occupy both sides of food webs. As predators, they capture plankton, larvae, and small animals, and in some cases can subdue prey larger than expected through repeated tentacle contact and venom delivery. This influences local community composition and energy transfer in pelagic and benthic systems. As prey, they are consumed by fish, turtles, sea slugs, starfish, and even other cnidarians, with some predators evolving behavioral or physiological tolerance to stings. Their ecological role also extends beyond direct feeding interactions. Reef-forming corals create three-dimensional habitat used by countless species for shelter, breeding, and foraging. So cnidarian influence is both trophic and structural. In freshwater habitats, hydrozoans contribute predation pressure on microfauna. In open marine systems, jelly-like forms participate in nutrient and energy pathways that remain active across depth gradients. This reinforces an essential point: cnidarians are not fringe organisms. They are recurring architects and regulators in aquatic ecosystems, shaping both who eats whom and where life can physically persist. Cnidarians are also a deep-time story. Fossils attributed to the phylum appear in rocks from around the Ediacaran period, roughly 580 million years ago, placing them before the Cambrian explosion. Coral-related records indicate major diversification trajectories extending back nearly 500 million years. Molecular clock analyses often suggest even older origins for the crown group, potentially around 741 million years, though estimates vary by method and dataset. What matters is convergence across lines of evidence that this lineage is ancient and persistent. Phylogenetic work supports cnidarians as a monophyletic group and positions them as sister to bilaterians, making them central to understanding early animal evolution. They are not simply survivors from an old world. They are active modern representatives of foundational animal design experiments that never stopped working. When you put all of this together, Cnidaria becomes a case study in durable innovation, a lean body architecture, cellular weaponry, flexible life cycles, distributed control, and ecological integration that has remained relevant from Precambrian seas to today's oceans. If you found this helpful, please like this video and subscribe for more explainers.